What if the most important medical discovery in history wasn't made by design, but completely by accident? In 1928, a scientist left his lab for vacation. When he returned, he found one of his Petri dishes contaminated with mold. Instead of throwing it away, he took a closer look, and that tiny speck of green fungus would change the world forever. This accident would lead to the creation of penicillin, the first true antibiotic, a discovery that would go on to save over 200 million lives and revolutionize medicine forever. But behind this miracle was chaos, war, and years of struggle, because the world almost never got penicillin at all. This is the unbelievable story of how mold accidentally saved humanity. To understand how miraculous penicillin really was, you first need to imagine what the world looked like before it existed. In the early 20th century, something as simple as a cut finger or sore throat could be deadly. There were no antibiotics, no pills, no injections to stop infections. A small scratch could lead to sepsis, and doctors could do little more than clean the wound and hope for the best. Pneumonia, tuberculosis, gonorrhea, strep throat, scarlet fever, and syphilis. These diseases killed millions every year. World War I had just ended, and infections claimed more lives on the battlefield than bullets. Medicine could mend bones and amputate limbs, but it couldn't stop bacteria. That all changed, because of one messy scientist and a moldy Petri dish. In September 1928, Dr. Alexander Fleming, a Scottish bacteriologist, was studying Staphylococcus bacteria at St. Mary's Hospital in London. He was known for being brilliant, but also incredibly untidy. When he left for a 2-week vacation, he left his Petri dishes unwashed on his desk. When he returned, he noticed something strange. One dish had grown a blue-green mold, but around that mold, the bacteria had completely disappeared. The mold seemed to be killing the bacteria. Fleming leaned closer, fascinated. He realized this fungus, later identified as Penicillium notatum, was releasing a substance that destroyed bacterial cells without harming human ones. He called this mysterious compound penicillin. In a single stroke of luck, he had stumbled upon something that could destroy infections without killing the patient, something medicine had been searching for since the dawn of civilization. Fleming knew he'd found something incredible, but convincing the world? That was another story. He published his findings in 1929, but no one really paid attention. His samples of penicillin were weak and unstable. They worked in Petri dishes, but he couldn't purify or mass-produce them. To the scientific community, penicillin seemed like just another failed experiment. For nearly a decade, penicillin sat forgotten, a discovery without a champion, until a new team came along and decided to make it real. In the late 1930s, at Oxford University, a team led by Howard Florey and Ernst Chain rediscovered Fleming's paper. They were intrigued. Could this mold actually become a usable drug? Along with a young biochemist named Norman Heatley, they worked day and night to extract and purify the active ingredient from the mold. The process was brutal. Each batch required hundreds of liters of moldy culture and days of work to get just a few drops of pure penicillin. At one point, when they ran out of material, Heatley even recycled the penicillin from patients' urine because the body excretes most of it unchanged. But then, it worked. When they tested penicillin on infected mice, every untreated mouse died. Every treated mouse lived. It was a miracle, and the team knew it. But as World War II loomed, they faced another impossible challenge: mass production. In 1941, Florey and Chain brought penicillin to the United States, hoping someone could produce it on a large scale. At first, no company wanted to take the risk. But when America entered World War II, everything changed. The government called on the biggest pharmaceutical companies, Pfizer, Merck, and Squibb, to help. Pfizer developed a deep fermentation process that used tanks instead of dishes to grow the mold. It worked, finally making penicillin in industrial quantities. By D-Day in 1944, Penicillin was being mass-produced and shipped to Allied soldiers across Europe. It prevented infections, saved wounded soldiers, and turned deadly injuries into survivable ones. The results were staggering. Mortality rates from infections dropped by over 90%. Penicillin didn't just save lives during the war, it changed medicine forever. After the war, penicillin became widely available to civilians. Diseases that once killed millions, strep throat, pneumonia, syphilis, were suddenly treatable. Fleming, Florey, and Chain shared the Nobel Prize in 1945, but Fleming never forgot how it all began. He warned that penicillin's power came with responsibility, that overuse could lead to bacterial resistance. And he was right. Today, antibiotics save lives every day, but antibiotic resistance has become one of medicine's biggest threats. Still, the story of penicillin reminds us of something timeless, that great discoveries often come from mistakes, and that even the smallest accident can change the course of history. Because sometimes, the world's greatest miracles start with something as ordinary as mold. Thanks so much for watching. If you enjoyed this story, don't forget to hit that like button and subscribe for more incredible tales from history. We'll see you in the next one.